produced by the host cell, so it's not actually a virus, and it always contains bacterial DNA, all right? Whereas in transduction, which is a way for horizontal gene transfer to take place, that's a viral particle. Some of those viruses have the viral genome inside of them. Some of them have a bacterial genome inside of them, all right? I think the other forms of horizontal gene transfer are relatively straightforward, but we're gonna hopefully get to those today, all right? Your other article that you read, I just really enjoy because it's a good example, I think, of environmental microbiology, right? And showing how two invasive species actually play off of each other and benefit from one another, right? So, you know, if you want to simplify it, say boar eat fungi, fungi gets in feces, seed finds feces, fertilizes seed so that it can grow, right? And normally that's a good thing, except when both species are invasive, then it allows them to spread to places they shouldn't go. So that's kind of a little bit of the summary of it. I'm really sorry that I can't hear you guys for some reason. Hopefully I can figure that out for my next class, or it'll be one-sided again. So please, if you have questions or want to interact, I guess we'll have to go through the chat box today. So my apologies for that. But let's go ahead and jump right in. So we talked about modifying nucleotides via chemicals. We talked about chemical analogs that resemble nucleotides, but are actually different in their hydrogen bonding capabilities. And so you get interesting base pairing going on there. And then another type of chemical mutation is something called, right, an intercalating agent. So if you remember intercalating from chemistry, right, it simply means, right, kind of blocking or displacing. So this is a physical displacement, right? And this physical displacement, what happens is it's kind of like I equate it to cutting in line, right? So when you cut in line, you shove your way between somebody, right? And when you shove in between somebody, well, then you're resulting in a frame shift, right? Or you're changing that 3 codon sequence that you're going to read. All right, and so some common chemicals that we see for this are ethidium bromide is a great example. We don't really use that as much in the lab at the undergraduate level because it's toxic. But what's really cool is it intercalates very well with DNA, and then you can hit it with UV light and you can see where your DNA bands are. So it's a really simple but powerful diagnostic tool. Chloroquine, for those of you that have maybe been to countries where malaria is an issue, you may have taken this. This is one of multiple reasons that if you're taking chloroquine for malaria, right, you're supposed to take it right before you go, while you're there, wherever you are, and then a little bit while you get back, but you're not supposed to stay on it for a prolonged period of time because it can have some negative effects on your DNA. And I can't remember, one group, I think Michael maybe and whoever Michael's presenting with, I can't remember, are doing malaria. And now anyway, we're seeing chloroquine resistant malaria. So yeah, down. Yeah, so the ethidium bromide, so basically the UV light is reacting with the ethidium bromide to give you that coloration pattern, right? So if you were to just hit a gel that didn't have ethidium bromide in it with UV light, you wouldn't see anything, all right? So what you're seeing is the ethidium bromide that's stuck to the DNA. Good question. Another type of way that we can induce mutations that's not a chemical induced mutation is radiation, right? And so when you hit with UV, when you hit DNA with the UV radiation, you cause dimers to form between thymines, all right? And so again, this would be like going outside without suntan lotion. You also have X-ray, right? X-rays is a type of radiation, and this induces strand breaks. So I don't have a picture that shows that, but think right here, right? You'd be breaking the backbone of the DNA, right? Now, if you break the backbone of DNA, it's very hard to replicate it, transcribe it, or translate it. When you have thymine, thymines that dimerize, it again causes misreading and confuses DNA polymerase and RNA polymerase, which is then going to cause a mutation, right? So this is what happens to your DNA if you're out in the sun too long without proper protection, right? And eventually things like this could lead to skin cancer. All right, and just a note too, for us, you'll see this different bacteria in just a minute. You can't fix this, right? So once this damage is done, it is done. So let's talk about repair. And a couple of these repair mechanisms that we're going to talk about right away in prokaryotic cells are very similar to repair mechanisms in your cells, all right? So, you can repair both spontaneous and chemical damage, all right? Just like prokaryotic cells can. And so one of the main ways to genetic repair is something called proofreading, right? And so this is actually occurring during replication, and it's a normal function of DNA polymerase. I don't know if you guys talked about proofreading that much in genetics. We talk about proofreading in genetics, Ajax? Briefly, you at least mentioned it, which is good. And so, right, DNA polymerase is laying down, right, new base pairs as it kind of reads the template strand of DNA, whichever way it's going, right, the leading or lagging strand. And while it does this, sometimes it makes mistakes. But what's really cool about DNA polymerase is it does have the ability to back up slightly, right, a few base pairs to fix any mistakes that it makes. And the way that it recognizes mistakes is that when you have base pairs that are bound together that shouldn't be bound together, it actually causes a physical distortion in that DNA. And DNA polymerase is able to detect or sense that physical distortion and then repair the base pair that is out of place. Okay. Another thing, right, that can happen is prokaryotic cells, and you have some too. You have a series of enzymes that can repair mismatches, just like we talked about. In this case, though, these are ones that DNA polymerase missed, right? This occurs after replication, right? So DNA polymerase has already gone off. And what these enzymes can do is they can cut out and replace a section of DNA. Sorry, I'll go back here. And so, here's your two strands of DNA. So let's say you lay down, right, your strand, and here's your mistake right here, right? You have a guanine binding with a thymine. So that's a bad thing, right? That's not what we necessarily want. And so in this case, right, this repair enzyme comes through, right, cuts out this strand, and then we lay down the correct nucleotides. Now, normally this is where I would ask you guys a question right before I did this, right? But since I can't hear you, it makes it a little complicated. So one of the things that we need to keep in mind, right, if you're looking, right, at these two anti-parallel strands, see here's the mistake. One of the questions I hope you're asking yourself is, well, how does the enzyme know that the mistake, right, is this thymine and not this guanine, right? So maybe this guanine should be an adenine, right? So how do we know where the mistake was made, right? And that's where methylation comes in place, right? So the longer DNA has been around, right, the longer chance we've had to methylate it. And so your body's kind of going on the premise, well, this is the methylated strand of DNA, so it's the template that's been around longer, and so we're going to strand. Now, it's true, right? If you make a mistake in the methylated strand, well, then you might have issues up here too, right? But that's kind of a quick little check, right? So the non-methylated strand is the new strand, and we say, okay, that's where the mistake is, and then we go and fix it. Now, this is where we get or where we diverge, I guess, from our prokaryotic brethren from back in the day, right? Is they have the ability to repair these UV damaged DNA, right, that causes these thymine dimers, right? So let's say you hit, right, prokaryotic DNA with UV radiation, right? You have these thymines that dimerize, they're covalently bound to one another. Right. So, right, they have a way, right, to protect themselves from this UV radiation, again, that you don't. And so we have something called light repair. Again, this is only in prokaryotic cells. And in light repair, you have enzymes that are activated in the presence of UV radiation, and they can come in, right, and break the covalent bonds that exist between thymine dimers, right? And so you actually return the strand back to normal, right? You don't replace any nucleotides. You don't have to bring in a polymerase. You simply break those covalent bonds between the thymine dimers and you move on. All right. Now, you also have, right? That's my good boy Snape, right? Master of the dark arts. You also have dark repair. And dark repair is also only in prokaryotic cells. And I guess you could kind of argue an advantage here is this is not dependent on enzymes that are activated in the presence of UV light. In fact, these enzymes work in the absence of UV light. And they are much more like the mismatch repair enzymes. They detect those thymine dimers. They remove nucleotides upstream and downstream, and then just replace everything. All right. Um, so, I guess the way you can think about this, if you've known someone that maybe has had a tumor, right, removed, right, some type of cancer surgery, if they can, they'll typically try to take a little bit of tissue, right, on either side, right, or tissue surrounding that tumor, even though some of it might be healthy tissue, to just make sure they get all of that tumor out. And that's basically what these enzymes are doing here, right? They're taking nucleotides on either side of that thymine dimer to make sure they properly remove the thymine dimer and then lay down these new base pairs. All right. And then my favorite repair that is only in prokaryotic cells is SOS repair, right? So SOS, you guys have probably heard of that. Save our ship. That's a distress call. That a sinking ship puts out, or sinking ships used to put out, and I assume, that applies to other forms of transportation now. But it's an extreme emergency, right? If you don't get here and do something quick, we're doomed. And so bacteria actually have an SOS system, right? That consists of something called SOS polymerase. And so what you just saw happen is basically that the DNA, right, or this chromosome of this prokaryotic cell just got hit with UV radiation, regular radiation, or X-ray radiation, chemicals, right? All of this fun stuff. And basically what this polymerase is going to come in and do is it works very quickly, and it does not have the ability to proofread, okay? So it would be like, let's see who I can see here. We'll do Ashlyn, right? It'd be like if I took Ashlyn and I put her in a boat and I poked a bunch of holes in the boat and it started sinking, and Ashlyn would just grab anything that's around her to plug into those holes, right? She's not going to analyze the structural integrity of substance 1, 2, 3, 4, and 5. She's going to take them. She's going to start filling those holes so that she doesn't sink. And that's what SOS polymerase does, right? It just starts laying down nucleotides as quickly as it can, right? Because the approach that the prokaryotic cell is taking here is it better to be wild type and dead, or is it better to be mutated and alive, right? Because a few mistakes have been made, right? So it chooses being a mutant and alive over being dead, right? Now, again, this would be normally this would be where I ask you, why is this not something that's good for us, right? And the reason this is not good for us is if you wanted, right, to do this system, right, the number of cancer uh, uh, diagnoses that we have, in the United States, in the world, would probably go up drastically, right? This would lead to a lot of issues. One of the main ones being potentially cancer in us, right? So this is not a good mechanism for us, but it is a good mechanism for an individual cell that is just trying to survive, right? And here, this is just a table, right, that kind of sums up some of those different repair mechanisms. Okay. Yes, Mikhail, SOS is only in prokaryotic cells. Only in prokaryotic cells. So what we're going to kind of talk about now in this next little bit are a couple different ways that we can identify mutants, and also how we can identify if a chemical agent is a mutagen, right? Meaning it's capable of inducing a mutation. Some of the phrases or terms that we're about to use, right, are something called an auxotroph, right, or auxotrophic bacteria, right? These require a growth factor, right, because they're they're no longer able to produce certain macromolecules, right? So basically, right, without the addition of an essential amino acid potentially, or without the addition of some certain type of fat or something like that, these bacteria won't grow, right? So auxotrophs, right, it's just a fancy word in this case for a mutant that we're studying. A prototroph, right, does not require any additional growth factors than are what are present in the media. So typically we refer to these as our wild type. Okay, so you'll see those terms come up a little bit later. So I just wanted to kind of lay the groundwork right there. So we have kind of two main ways that we can select bacterial mutants. We can do that through direct selection, which we're going to talk about right here. We can also do that through indirect selection, which we'll talk about in just a second. Now, in direct selection, right, one of the simple ways to think about this is these bacteria grow where their parents can't. So in this case right here, right, you have, think of it like TSB, right? So a media or broth that can grow anything. Inside of that media or broth, you have streptomycin resistant cells, which are the mutants. You have streptomycin sensitive cells, which are a wild type cell. And so what you do is you take a little bit of liquid, right, from this culture. You put it on a plate that has streptomycin in it, and then you grow them on a plate that has no streptomycin. And here without streptomycin, just like in this tube of broth, you get a large growth, a bunch of different colonies. However, here, right, you get only your mutant growing, right? So you select it directly, right? You can see it grows on the plate with streptomycin. So if it grows, you automatically know it's a mutant. That's all you have to do. Okay? Keep that in mind, right, as we go to indirect selection. So in indirect selection, again, in this case, you have a plate that has your auxotrophic bacteria and your prototrophic bacteria growing on it, right? It's a mix of different colonies. You don't know which is which because this plate, this nutrient auger, contains everything that you need. What kind of question would you ask in the exam about this topic? Which topic? The repair? Is that what you're talking about, Holmes? Yeah. So, I mean, I would ask probably how prokaryotic cells are able These two slides, direct and indirect. I would ask how you select mutants, and I could ask you to walk me through uh, the process or how they work or how they select. So in this case, with indirect selection, right, you have your wild type and your mutants growing on this plate. It's a nutrient auger, so that means it has all the required nutrients, even for a mutant to grow. So you can't distinguish who's a mutant and who's not a mutant. You then basically create a stamped copy of your bacteria. So in this case, it's a sterile velvet. You press it against the plate and you transfer over all of those colonies. So you're basically making an exact replica of the bacteria that are growing on that plate. You then take that stamp and you press it onto two different types of media. One is the same media that you had before. It's the nutrient auger. It contains everything. So every bacteria on that stamp should be able to grow. Here you have something called your glucose salt auger, right? You could also call it minimal media. And basically what that means is the only thing it has is glucose and salts. If you're a wild type bacteria, this is enough for you to make all of the macromolecules that you need, right? Remember from previous biology classes, you could take something that is a carbohydrate and you could break it down and then rearrange it to make an amino acid. You could break it down and rearrange it to make a fat, all right? And so if you're a wild type bacteria, right, you can grow really well on this plate. If you're a mutant though, right, this is lacking specific nutrients, you can't grow. So then what you do is you compare your two plates, right? So this is why it's indirect. So you look at your plate where everything grew, and you look at your plate where you now have where you have your minimal media, and you see, okay, well, there's a colony there on my nutrient auger, but it's missing from my minimal auger. So then I can go back to this plate, even though it has all these colonies growing on it, and I can say that is my mutant, all right? So that's why it's indirect, right? You're actually selecting it from an auger that grows everything by comparing it back to an auger that can only grow a few things, right? Please look back through this. It's not, again, I don't think this is an overly complicated method, but you might need to review it just to make sure you're getting those different steps right. Another thing, right, that we can do when it comes to indirect selection is use something called penicillin enrichment, right? So again, I'm not going to make you guys answer these because I can't hear you right now for whatever reason. But basically, right, what do wild type cells do in minimal media? Well, they divide, all right? What do mutant cells not do in minimal media? Well, they don't divide because they don't have the nutrients that they need. And so what you can do, right, is you can take penicillin and you can actually add it to your minimal media broth, and it kills those cells that are dividing, all right? And so that means when you culture this broth on a plate, you're going to have more of your mutants growing than you actually are your wild type, right? And then with this plate, you would still go through the process of indirect selection. It would just be much easier to identify your mutants, right? Because you wouldn't have as many wild type interspersed amongst those mutants. So penicillin enrichment, you still have to go through indirect selection, but it changes the ratio in a positive way, right? It gives you more mutants present, removes a bunch of the wild type, and makes it much more easy. It makes it much easier for you guys to identify those different cell types. Okay. So that was how you select mutants, right? Direct and indirect selection. Now, how on a prokaryotic level do you identify a mutagen? And one of the main ways that you do this is something called the Ames test, all right? Commonly for this test, we use a histidine requiring auxotroph, right? This is Salmonella typhimurium. So basically, this is a fancy way of saying salmonella, this strain of salmonella will not grow unless you add histidine to the plate, all right? So a glucose salt agar plate won't support growth of this salmonella mutant, all right? You also see here, and I'll point this out in just a second, that in order to more accurately identify a mutagen, sometimes we mix that mutagen with animal liver extract. The reason that is some of you that are going on to be PAs, maybe even pharmacists, doctors, something in the medical field, right? One of the things you need to appreciate and be aware of is sometimes, or oftentimes when you take medication, those medications will go through the liver where they can potentially be modified, right? And sometimes that modification could cause something that wasn't a mutagen to become a mutagen. And so by adding liver enzymes to whatever chemical we're studying, it might give us a clearer picture as to whether it's a mutagen, right? So let's talk about this quick. Again, relatively straightforward. So here you have your glucose salt, right, media, or your minimal media. It's lacking histidine. So if I put my salmonella on it, right, I would expect nothing to happen. I then also, because I'm doing a good experiment, have a control liquid, right? So this liquid is basically, however I plan to deliver the mutagen, this is the liquid without the mutagen. So you have a control liquid, you have your mutant bacteria, you put them together, and you incubate it. Now, you see a couple colonies that grow here, but this is because, again, of a spontaneous mutation. Remember, bacteria, prokaryotic cells, are able to revert back to their wild type form. So you expect that to maybe happen, all right? So this is a really important control plate to do, because then you do your real experimental plate. So again, you have your minimal media plate, you have your bacteria that can't grow without histidine, you add your suspected mutagen. This would also be the point where you add your liver enzyme extract, if you're doing that, which would give you a little bit more of an accurate picture. And then you incubate them, all right? And here, right, you see if the chemical is a mutagen, you have a lot of colonies that grow, right? You induced a reversion in a lot of those salmonella bacteria that couldn't grow before. If it's not a mutagen, right, you see here that you just have a couple colonies that grow. And this is why it's really important to do that control. If you didn't do this control here on the left, you'd have no idea officially if these bacteria spontaneously reverted or if the reversion was induced by a mutagen. So you always need to do this control plate to compare back to your mutagen, right, to make sure that you're not looking at a spontaneous reversion, you're looking at an induced reversion. Okay? Cool. So horizontal gene transfer, right? This is what we're going to talk about here for the next little bit. And I think that'll be as far as we get, which is fine, because hopefully you've read that article. If you haven't read the article, right, you have a little bit to another day to read that before your exam coming up, right? So here, right, horizontal gene transfer is cell-to-cell DNA movement, all right? In this case, right, we would say that DNA is kept by the recipient cell as it divides, right? So this is an effect that is not just one generation, right? This DNA is incorporated into the genome and then is part of all subsequent cells that come off of that mother cell, all right? Primarily, what we'll talk about is homologous recombination, right? So this is exchange of DNA between similar regions, right? A relatively conserved, process. And so again, when you're adding a gene, it is filling the space that was occupied by a similar gene. We may get to, but probably won't talk too much about non-homologous direct insertion, but an example of this would be those transposable elements that we talked about earlier, right? So when the gene inserts, it's inserting itself perhaps in a sequence that has no similar genes or nothing to do with that gene that's being transferred. So from your paper, hopefully these are familiar: transformation, transduction, and conjugation, right? All ways that genetic information can be shared between prokaryotic cells, right? And then we had the gene transfer agents, which are not near as common as these three modes of horizontal gene transfer, but still important, right? So let's talk about these, really briefly. So what are some of the important characteristics of transformation, right? So these cells are taking up naked DNA. Now the DNA is out there because it has either been secreted by a cell, right? And this can be a defense mechanism. A cell knows that it's going to die. It knows that it is under attack, and so it can secrete its DNA. It can also be the result of cell lysis, right? So a bunch of bacteria die, they're killed off maybe by an antibiotic, right, as an example. And they start to break down and their DNA gets released. Now, what's really important, right, is that cells have to be competent in order for this DNA to be taken up, right? So what that means is if Alanis's favorite animal is a hedgehog, am I close? Good. Yeah, yeah. Alanis, just right. And so if I took a bunch of hedgehog DNA, right, and we won't talk about how I got all this hedgehog DNA, but let's say I had it and I dumped it on Alanis, all right, Alanis would not turn into a hedgehog. Alanis's cells are not competent to take up random segments of DNA, right? And so that's actually a really special feature, right, to be competent. All right. What we see is that cells that are super stressed can upregulate receptors that make them competent, right, so that they can receive DNA, right? So although it shows this DNA just entering the cell randomly, it's not exactly how it works, right? There are specialized receptors that bind to this DNA and help it gain entry into the cell, right? We see some bacteria are just better at this than other bacteria, so not all bacteria are capable of being competent, and some bacteria are only competent when it's under stress, right? Stress or duress. All right. And again, tightly controlled population density and nutrient availability. So again, you're not going to see this as much if everybody's happy and things are going well. But if population is growing rapidly, nutrients are being depleted, then you might see some cells become more competent, right? And then they can take in this DNA, and this is talking about, right, and the daughter cells have to have it. So once it's incorporated into your genome, when you divide, the daughter cell has it. When the daughter cell divides, the next daughter, right? So it keeps on going down from generation. So it's permanently becoming part of this genome. You also have transduction, which again, I'm not going to spend a lot of time on this process here because, right, you guys already know how this happens. So we follow the regular infectious cycle of a bacteriophage, right? So it comes in here, slices up the chromosome, right? We package and we make new viruses. The problem here, I guess if you want to call it a problem, is that sometimes the virus makes a mistake. It accidentally packs, you can see here in the purple, the segment of bacterial DNA. We call this a transducing particle because it now has bacterial DNA. So that transducing particle basically is a virus on the outside and a little bit of bacteria on the inside. And so when it goes on to infect a cell, right, like a virus does, right, it attaches, and instead of inserting viral DNA, it inserts bacterial DNA. And then that bacterial DNA finds the homologous area of that bacterial chromosome and replaces the existing host DNA, right? And now you have, right, a slightly altered bacterial chromosome. And again, one of the biggest things that we fear or we get concerned about in horizontal gene transfer, and I talked about it in the article that you guys hopefully have read or will read, right, is antibiotic resistance, right? So this is a big deal, right? Information that's detrimental to us could be shared between bacteria. All right. Here you have conjugation, right, which we've already mentioned briefly when we talked about fimbriae. right? And so this is bacterial sex, right? Or as close, right, to sex as bacteria get. And so in this case, you can have plasmid transfer or chromosome transfer. This would also be the point where I would ask you what's more likely to be transferred. And you would probably say that a plasmid is more likely to be transferred, and I would say that you are correct, right? One of the things that you don't appreciate is that conjugation is typically, or that you often don't appreciate, I know I don't appreciate enough, is that conjugation is taking place in an unstable environment, all right? And so this connection that's formed between these two bacterial cells is actually relatively unstable. Plasmids are a lot smaller than chromosomes, and so it's easier to get a plasmid across this F pilus before this connection is broken compared to trying to move a whole chromosome. But saying that, it is possible for an entire chromosome to be transferred. All right. So this is what that process looks like. Again, cells that are capable of conjugation have something called an F plasmid, right, or fertility plasmid, which allows them to create a fertility pilus, right? So that's what the F plus is. The recipient cell is F negative. It does not have that plasmid or that fertility pilus, right? A connection is formed via that pilus, right? So this is the process of conjugation. We then start to replicate, in this case, a plasmid. As it's replicating, one strand, right, moves across, right? As you move that strand across, you immediately start to add the corresponding base pairs. And then at the end of the process, you're left with two F plus, right, fertility positive cells, all right? Again, I said this can happen with chromosomes. Even if you only get part of a chromosome across, right, then you could replace the homologous section of the recipient chromosome also. Okay? So again, right, plasmids, there can be a lot of plasmids in a cell, right? Cells can have anywhere from, they could actually have 0 to 500 copies of a plasmid, right? So again, a lot of different DNA, right? Host range, so I say specific or broad. What that means is that different species of bacteria can actually share, in some cases, share plasmids, while some plasmids are only shared amongst bacteria in the same species, all right? Right? These can, these plasmids or other plasmids can be transferred, just like the F plasmid can be transferred, right? But you still are going to need that F plasmid to initiate that conjugation event. And then we have something, right, called an R plasmid, which is a plasmid for resistance. And then you see on this plasmid, it actually has 3 genes for being resistant to a certain antibiotic. Okay, so this can be a huge, huge issue just across the board for us, right? Horizontal gene transfer is a really kind of, I would say, unique survival mechanism, right? And so it could be really important for helping a bacteria make an enzyme that allows them to utilize a different nutrient, right? Which is awesome for their survival. And that's not really our big concern, right? We become concerned about horizontal gene transfer when they're sharing information that allows them to be resistant to drugs, all right? You know, what we are doing and what we are utilizing bacteria for in some cases is something called genomic islands. And so here you're taking a large segment of DNA from a cell's genome that originated in one species and putting it into a new species. So an example here, right, would be Bacillus thuringiensis. This bacteria is actually kind of a natural pesticide. And so what we've done is we've taken that segment of the genome from this bacteria and put it into corn. And so now you have corn that produces a natural antibiotic. Again, I'm not here to debate the ethical implications, I guess, of genetically modified organisms or GMOs as a food source, but I'm just saying this is something that we're doing. And one that's near and dear to my heart, having done some diabetes research, is that we've actually taken the human gene for insulin, right? So this is actually insulin that's being produced right here, and popped it into E. coli. And since E. coli are very easy to grow, which you guys can appreciate from the lab, we have an easy source of insulin, right? So we're basically making the E. coli produce insulin for us, and we can then isolate it from that culture, right? Again, you guys can look over these terms. You probably have heard these before, but genome is a complete set of genetic information, right? So like a microbiome is all of the genetic information from the microbes in you. Gene, we've talked about it as a functional unit. And so these are just some things that you may see in the future, all right? And that's it, okay? So your exam will be Friday. Up to this point, please look through those papers. If you have not, don't forget I have a, oh yes, study session. Thank you, Micaiah, for bringing that up in the last couple minutes, or reminding me of that. So Ajax, I think, is willing to do a study session tonight. Ooh, here, hold on one second. Hello. I'm doing well. How are you? Okay. So I am clear to return to action. I still have a little bit. I still have a little bit of like a stuffy nose, but it's gotten a lot better. Correct. Yes, ma'am. Yes, ma'am. No, I mean, I haven't taken my temperature, but I haven't felt feverish. So awesome. Oh, no, 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 no. That's totally fine. I really appreciate that. And do I need to, so is it okay? Do I need to have you send something to Dr. De La Luz, or is it okay if I just tell him?